Before we begin, I would like you to understand one thing. These videos are created based on information taken from scientific articles or texts written by professionals. In the video description, you can find the main bibliographic sources I have used, so you have the opportunity to read them more thoroughly and avoid any misunderstandings. Additionally, I encourage all professionals in the field to correct and comment on the videos in a polite manner if they hear any incorrect information, in order to create a constructive and productive discussion on each topic covered. I wish you a pleasant viewing. The gut microbiota, a hidden ecosystem that regulates the body's health. Have you ever imagined that an entire universe of microorganisms hides in your gut, capable of influencing your health? Let's explore this hidden organ and its fundamental role in our well-being. Our gut hosts a complex ecosystem of microorganisms, about 250 species of bacteria, viruses, fungi, and archaea, that together form the gut microbiota. This collection of microorganisms performs vital functions, from digestion and nutrient synthesis to immune system modulation and brain communication. The most prevalent species include 1. Firmicutes, includes genera such as Lactobacillus, Clostridium, Enterococcus, and Ruminococcus. These bacteria are essential for carbohydrate fermentation and butyrate production. 2. Bacteroidetes. This group includes several classes involved in nutrient metabolism. 3. Other species such as Vericomicrobia, Actinobacteria, Proteobacteria, and Archaea. This composition varies over time, influenced by factors such as diet, age, lifestyle, and environment. What are the key functions of the microbiota? 1. Digestion and metabolism. The microbiota doesn't just reside in our gut. It acts as an internal laboratory that transforms what we eat into energy and essential substances. It breaks down complex carbohydrates. The microbiota decomposes polysaccharides and indigestible fibers into short-chain fatty acids, SCFAs, such as butyrate, acetate, and propionate. These compounds provide energy to intestinal cells and help regulate inflammation. Synthesizes essential nutrients. Besides SCFAs, microorganisms produce vitamins such as vitamin K, thiamine, folate, riboflavin, pantothenic acid, and biotin, and some amino acids supplementing our diet. 2. Protection and defense. The microbiota acts as a barrier and ally of the immune system, protecting our body from pathogens. It supports epithelial cells and mucus that cover the intestine, preventing pathogen entry. It enables immune system maturation. By interacting with immune cells, the microbiota helps train the immune system, making it more efficient in distinguishing between harmful and harmless agents. 3. Communication between organs. The microbiota communicates with various body parts, influencing processes far from the intestine. For example, gut-brain axis. The microbiota produces metabolites and neurotransmitters that affect mood, behavior, and cognitive function. Many mental disorders appear to be linked to this axis. Gut immune axis. It regulates immune responses and maintains the balance between pro and anti-inflammatory reactions. Gut-liver axis. The portal circulation transports bacteria and metabolites to the liver, influencing metabolic processes and potentially contributing to liver diseases. There are also connections with the entire body. The impact of gut microbiota on health. Key metabolites and inflammatory diseases. The gut microbiota is not just a collection of microorganisms, but a true ecosystem that affects our health. When its composition changes, so does the metabolome, the set of metabolites produced by bacteria, leading to direct consequences on intestinal well-being. This phenomenon is particularly evident in chronic inflammatory bowel diseases, IBD, such as Crohn's disease and ulcerative colitis. The main substances produced by these microorganisms to protect us include 1. Short-chain fatty acids, SCFAs. SCFAs are key metabolites produced by the fermentation of dietary fibers by gut bacteria. The most important ones include butyrate, acetate, propionate. Butyrate concentrates mainly in the colon, where it plays a crucial role in nourishing intestinal cells and modulating inflammation. SCFAs can cross intestinal cells, interact with the body, and regulate the immune system. A higher fiber intake increases the production of these SCFAs by the microbiota. A fiber-rich diet promotes the growth of beneficial bacteria such as Prevotella, Xylanobacter, Faecalibacterium prosnitzi, one of the main butyrate producers. A higher presence of Faecalibacterium prosnitzi is associated with reduced intestinal inflammation, whereas its deficiency has been observed in Crohn's disease patients. 2. Bile acids. Bile acids not only aid in fat digestion, but also play a key role in metabolism, immune response, and the regulation of numerous physiological processes. But how are they formed? The liver produces primary bile acids from cholesterol. These are released into the small intestine to aid digestion. The microbiota converts primary bile acids into secondary bile acids through enzymatic processes. Bile acids are then reabsorbed and recycled in the liver. Their function will be metabolic regulation. Specifically, they influence fat and glucose metabolism through specific receptors such as FXR and TGR5. Inflammation control. They modulate the intestinal immune response. In IBD, bile acid metabolism is altered due to intestinal dysbiosis, leading to a reduction in secondary bile acids, an increase in primary bile acids, which are more irritating to the intestinal mucosa, a microbiota imbalance, with the growth of bacteria such as proteobacteria and fusobacteria at the expense of firmicutes. Inflammation of the ileum, typical of Crohn's disease, reduces the transformation of bile acids, worsening metabolic imbalance and dysbiosis. 3. Tryptophan. Tryptophan is an essential amino acid that influences both the nervous and immune systems. It can follow 3 metabolic pathways. The kynurenine, or serotonin, pathway. Through this pathway, tryptophan can be converted into serotonin and melatonin, regulating mood and sleep. Alternatively, it can be transformed into nicotinamide, vitamin B3, which is essential for energy metabolism and cellular repair. The indole pathway. Certain gut bacteria convert tryptophan into indole derivatives, such as indole acetic acid and indole 3-aldehyde. These compounds protect the intestinal barrier, regulate the immune system, and reduce inflammation. The tryptamine pathway. Some bacteria, such as Clostridium sporogenes, convert tryptophan into tryptamine, a compound that interacts with the enteric nervous system and modulates intestinal motility. Its role has been hypothesized in irritable bowel syndrome, IBS, and intestinal secretion regulation. Indole metabolites, in particular, activate the aryl hydrocarbon receptor, AHR, which has a strong anti-inflammatory effect. Even a tryptophan deficiency has been linked to worsening ulcerative colitis and Crohn's disease. But be careful. Under normal conditions, there is a cooperative relationship between the host and microbiota, known as symbiosis. In this relationship, the host, us, provides a stable environment and nutrients, while the microbiota supports numerous physiological processes. An alteration in microbial composition, known as dysbiosis, manifests as reduced microbial diversity, a decrease in good bacteria, and an increase in potentially harmful species. Increased pro-inflammatory components. For example, an increase in gram-negative bacteria can lead to higher amounts of lipopolysaccharide LPS, a component of gram-negative bacterial membranes that strongly triggers inflammation. Its binding to toll-like receptors, TLRs, particularly TLR4, can activate a signaling cascade leading to excessive cytokine production and, in some cases, intestinal fibrosis. In dysbiosis conditions, chain effects can occur, leading to gastrointestinal and systemic disorders. Dysbiosis has been linked to inflammatory bowel diseases, IBD, irritable bowel syndrome, IBS, metabolic disorders such as diabetes and obesity, allergies, and even autoimmune diseases. But how does this alteration manifest? Recently, some fascinating discoveries have been made. Depending on the predominant microorganism, different types of dysbiosis have been identified, including 1. SIBO, small intestinal bacterial overgrowth, in excess of bacteria normally found in the colon colonizes the small intestine, causing abnormal fermentation. 2. LIBO, large intestinal bacterial overgrowth similar to SIBO, but localized in the colon, leading to alterations in intestinal motility. 3. SIFO, small intestinal fungal overgrowth, involves fungi, such as Candida, causing gastrointestinal symptoms similar to SIBO. 4. IMO, intestinal methanogen overgrowth, not a true bacterial overgrowth, but an increase in methanogenic microorganisms, such as Methanobrevibacter smithii, which slows intestinal motility and promotes constipation. Among these conditions, SIBO is the most common, even found in patients without known diseases. The bacteria involved, including Streptococcus, Staphylococcus, Bacteroides, and Lactobacillus, proliferate in an abnormal location, leading to early carbohydrate fermentation and gas production, such as hydrogen and methane. The result? Bloating and abdominal distension due to gas accumulation. Altered intestinal motility with diarrhea or constipation depending on the type of gas produced. Abdominal pain and cramps, similar to those in IBS and IBD. Moreover, SIBO has often been found in chronic and systemic diseases, including non-alcoholic fatty liver disease, liver cirrhosis, chronic pancreatitis, obesity, cystic fibrosis, and much more. This correlation suggests a deep connection between the microbiota and systemic health, opening new research perspectives. How is SIBO diagnosed? Non-invasive tests such as the lactulose breath test, LBT, are used to measure hydrogen and methane production after lactulose ingestion. This sugar, non-absorbable by the body, reaches the small intestine intact, where it undergoes fermentation if bacterial overgrowth is present. An alternative is the glucose breath test, GBT, which uses glucose as a fermentable substrate, but has the limitation of detecting bacterial growth only in the early portions of the small intestine. How can SIBO be managed? Since SIBO is fueled by bacterial fermentation, an effective strategy is to reduce the intake of fermentable carbohydrates, such as those found in legumes, onions, garlic, and cabbage, rich in oligosaccharides, milk and dairy products due to lactose, excess fructose, found in certain fruits and sweeteners, polyols, such as sorbitol and mannitol. This approach, known as the low FODMAP diet, reduces fermentation and symptoms. However, it must be managed carefully by a healthcare professional to avoid nutritional deficiencies. Antibiotics, while necessary in many situations, can profoundly alter microbiota balance, with both short and long-term effects. Therefore, always let healthcare professionals guide their use with proper awareness, as research has advanced significantly. The gut microbiota is a fascinating world that profoundly influences our health. We've seen how crucial it is to maintain balance through diet and lifestyle. And you? Have you ever experienced intestinal disorders or adopted strategies to take care of your microbiota? Share your experience in the comments. If you enjoyed this video, leave a like, share it with colleagues and friends to spread this information, and subscribe to the channel so you don't miss future content on nutrition and well-being. See you soon.